Hello dear students, today we will discuss about the detection of viral growth in cell line. In the previous lecture we discussed that what is cell line and how we can culture the virus. Now after culturing the virus in a specific cell line, how we detect viral growth. There are various types of methods which we use for the detection of virus in a cell line or cell culture. In this lecture, we will explain what is cytopathic effect, viral interference, inclusion body, detection uh, to detect viral antigens by direct immunofluorescent assay, immunoperoxidase staining, hem absorption. We will also discuss electron microscopy. Electron microscopy is used to detect viral particle in the infected cell lines. Viral gene detection by using PCR or nucleic acid probes. So, first of all, we are going to discuss what is cytopathic effect. It is defined as the morphological change morphological change produced by a virus in the cell line detected by light microscope. When you culture a virus, this what this virus do actually, suppose if this is a cell, the virus will infect this cell. It will this virus will produce some morphological changes in the cell. They will round the cell or they uh, shrink the cell, etc. We will detect these changes by using light microscope, and these changes are called cytopathic effect. Cyto means cell, pathic means disease. It will cause disease in the cell in the form, and they will change its morphology or shape. This change in the this morphological change in the cell caused by virus is known as cytopathic effect, and it help the virologist to find out whether the virus is present in the cell culture or not. You should note that not all virions have the ability to replicate in the host cells. It should be noted. Those virions that have this ability are said to be infective, and the term infectivity is used to denote the capacity of virus to replicate. Virus may be non-infective because why? Because they lack part of the genome or because they have been damaged. To determine whether a sample or a specimen contain infective virus, it can be inoculated into a culture of cells. It means that suppose if a sample, if we know that either it is affected by a virus or not, what we do, we inoculate a virus, we can inoculate a virus into a culture cell or a host organism for to know to support the replication of the virus suspected for being present. Now what happens when you inoculate the virus to a cell? After inoculation you incubate. Incubate mean to provide specific temperature. So you should incubate the cell culture at appropriate temperature. It can be examined by light microscope for the characteristic changes in the appearance of the cells resulting from virus induced cell. It means that when you want to locate the virus, what what should do? You should pick the cells, then you inoculate the cells, and after inoculation, you examine this cell by which type of microscope? Light microscope. Now, a change of this type is known as cytopathic effect. The virus-induced damage will be known as cytopathic effect. Now, different viruses show different effect. For example, poliovirus infected cells have shrunk and become rounded, while a multinucleated giant cell known as syncytium has been formed by the fusion of membrane in the case of herpes virus. Poliovirus, they will shrink the cell. They will shrink the cell, and the herpes virus, they will fuse the cell. They will fuse the cell, and this fused cell is known as syncytia. If we look at these figures, A and figure A, cytopathic effect caused by the replication of poliovirus and herpes virus are shown in these figures. This show uninfected cell, while the B it show the infected with polio cell. What polio? Actually, polio viruses have shrunk the cells. Look at these cells; they have shrunk the cells. Now, in the case of herpes viruses, these are non-infected cell, but these are affected cell. The affected cell are fused together and form a structure that is called syncytia. Now, type of cytopathic effect. Now, what enterovirus do? Enteroviruses, they will cause the degeneration of the entire cell sheet. They will, suppose this is the cell when affected by a virus, it will degenerate. It will degenerate. Measles virus, respiratory syncytial virus, and herpes simplex virus, they what they do? They form giant cell formation. They fuse all the cell together and they form syncytia. This is called syncytia. Now, herpes simplex virus actually diffuse rounding and ballooning of the cell line. What they do? Herpes simplex virus, they rounding the cell like a balloon in the cell line. Now, simian vacuolating virus 40, what they do? Cytoplasmic vacuolation. They form, suppose in this cytoplasm, they form rounded structure, and that is just like vacuoles. What adenovirus do? Adenovirus, they actually produce grapes like structure. They produce grapes like structures. Now, what is viral interference? The next topic is viral interference. What does it mean? The growth of cytopathic effect virus in a cell culture can be detected by the subsequent challenge to the cell line with a known cytopathic effect. Suppose if a virus is do not show cytopathic effect in the cell culture, how we can detect? We can detect it by changing the cell line with cytopathic virus. The growth of the first virus inhibit infection by the second virus. The first virus, suppose what they will do, the growth of this virus is inhibited by this second virus. They will inhibit its growth. For example, rubella do not show cytopathic effect. uh, what will do but prevent the replication of enteroviruses. It will do which are known to produce cytopathic effect. So enterovirus cytopathic effect can be inhibited by rubella virus. This phenomena is known as viral interference. Now, the next is inclusion bodies. What are inclusion bodies? Actually, inclusion body are aggregates of virions or viral particles or their products during its replication, which can be demonstrated in virus infected cell under the light microscope. Inclusion body, these are small aggregate of virus. These aggregate may be formed from the viral protein. Actually, they may be other product during its replication, and these aggregate can be seen through the light microscope. Now, different viruses produce different inclusion bodies. They may be inclusion body may be produced in cytoplasm, intracytoplasmic inclusion bodies. They may be intranuclear inclusion bodies. Now, intracytoplasmic inclusion bodies, you should note rabies virus. Rabies virus attack on the central nervous system, and they produce special type of negri bodies. When we can diagnose if a brain or central nervous system contain the negri bodies, it will be caused by rabies virus. Pus gene bodies seen in the varroa virus. Uh, Gornary bodies seen in the Vaccinia virus, Bollinger's bodies seen in the Pallfox virus, molluscum bodies seen in the molluscum contagiosum virus, and perinuclear cytoplasmic bodies seen in the Riu viruses. Now, some viruses produce inclusion body in the nuclear. For example, cowdery type A inclusion, the Taurus body seen in the Yellowpurus virus, Lipschultz body seen in the Herplex virus, Cowdery B-type inclusion seen in the poliovirus. Non-specific may be lesser type. Intracytoplasmic intranuclear inclusion body seen in the measles and cytomegalovirus. It means that cytomegalovirus and measles produce intracytoplasmic and intra inclusion bodies, and these are called oil eye appearance. They look like the eyes of a oils.